Hello friends, today we are going to finish this actual morphology part with the seed structure. Already we have learnt about fruits and fruit types. So actually you must be aware about how the seed must be taking place. So how the seed will develop, how the seed must be taking its position in the fruit. Okay, what is seed? Seed is nothing but it is fertilized ovule. When we say a pollen grain falls there on the stigma, that time it forms a pollen tube, and after forming a pollen tube reaches to the egg cell, that is site of fertilization, and after fertilization it forms an embryo. Simultaneously, embryo needs some nourishment, some food, some additional nutrient elements. Those elements are always provided by the endosperm. How endosperm is formed? Endosperm is formed as a result of double fertilization. So deliberately I am using this word double fertilization. Okay. So this particular topic, that is study of fruit and seed part 2, is all about study of seed. Okay. Now you should understand why we have means humans, we have generated so much of interest in this particular seed and seed behavior, because whatever the germination occurs, whenever the regeneration occurs, whenever the reproduction shows its results, that time seed behavior and seed structure, that always matters a lot. Means when we talk about a typical seed, let there be monocot or dicot, we are considering that all the genetic information about a plant is stored there with the small sized seed. This particular seed, it always gives rise to new plant after germination. How germination occurs? Germination occurs when suitable and favorable conditions are available, enough moisture, enough water and enough warmth. If it is available, then that particular germination is possible. Now, I have a couple of points to discuss with you, but I would like to tell you all the points which we are going to consider. First of all, why there are no seeds in animals. This will be the first point. And second point is that why animals, they always show what I can say, independent behavior with respect to the sexual life. Now I would like to comment on the very first point. This particular animal world is movable. They never station themselves at one place. They never spend their entire life at one space, at one point. That happens in case of plant world. Plants, they stay there. Hence, they require some additional mechanism to enhance the chances of growth, means spread. For example, in case of dogs and cats and humans, in case of animals, they are locomotory. They can move from one place to another place. Hence, after fertilization, there is formation of zygote, there is development of embryo, and baby takes a birth. So this is the normal cycle in any animal case. Here in case of plants, we have seeds. Why do we have seeds? Because there is a dormancy period. There is a word dormancy. Do you understand why the word dormancy? Suppose this year on 31st of December, if any plant produces a seed, that seed will remain dormant for say 5 years. Means within the period of 5 years, if you sow that seed in appropriate climatic condition or favorable conditions, growing conditions, then definitely that seed will germinate. When that seed will germinate, that time you can say, okay, new plant is growing, and eventually this plant is going to develop like a huge tree, from a smallest seed. So this happens with this particular plant kingdom. In case of animal kingdom, we don't need any seed. We don't need any dormancy period. It is not like that we are bound somewhere in the cocoon and at favorable time we come out from the cocoon. It is not like that. Actually, whenever copulation occurs, whenever there is fusion of egg cell and sperm cell, that time the zygote formation occurs, zygote divides and redivides and it forms embryo and further it forms a baby. So this particular mechanism is applicable there in animals only, because animals are locomotory. They can move from one place to another place. Now once again think about the plants. One plant is placed at this place, one plant is situated at another place, and if cross pollination occurs, then, or let there be self pollination, don't worry about the pollination mechanism. When seed setting occurs, you cannot expect that immediately that seed will get a favorable condition. Unless and until that seed gets favorable condition, it will not germinate. There are several chemical reactions, there are several hormonal actions which are taking place in regular seed life. Yes, I am using the word seed life. Life of seed begins right from the seed formation, seed setting. That particular word is seed setting. Right from seed setting up to the maturity of seed. Then maturity of seed up to the dehiscence of fruit. If fruit is non-dehiscent type, then definitely it will fall down and the outer part it will degenerate. It will be consumed by some animals or some insects, and rest of the things which is there, which is protected in some cases. So definitely the seeds will become open, and after favorable condition they will germinate and they will form an entire new plant. So this happens with the plants only. Another point I have to say, purposely I am writing all the points very carefully on the piece of paper. This helps me to remember each and everything. For you people, we also suggest that you should also have a small pocket diary. Actually, it is given to you along with your study kit. So you should have it regularly. Whenever you think about anything, you should write. You should write it and you should refer it after 2 hours or 3 hours or at the end of the day. So you can refer it. So it will help you to organize yourself while carrying out any exercise, while carrying out any study performances. So that will help you a lot. We have done with this why there are no seeds in animals. The point, one more I would like to tell you, that embryo of animals are not encapsulated, and the embryo of plants, they are capsulated. Here in case of animals, as I mentioned, when we talk about fertilization process, egg cell, it is getting fertilized by the sperm cell, and ultimately the zygote formation occurs. Then embryo, it is present. Embryo grows there in the womb of a female, and embryo gets continuous food supply. Okay, there are several arrangements. Then that embryo that takes a birth after gestation period is over, and that independent individual can grow directly and it can develop itself. Okay, in case of plants, if embryo, if it is not getting favorable conditions immediately, then definitely the thing should be protected. This actually arrangement is done by nature. So whenever we talk about human development and achievements of human, we have done nothing. We have simply used the technology. We don't have that creativity. We have creativity, but we don't have that creativity which nature has. Okay, so always focus on the nature's creativity. So this particular seed structure is encapsulated. this particular seed structure is encapsulated. Seed actually contains embryo. So this embryo, so further it develops and then it develops into a new plant. Now it's a time for we people to understand how seed develops. Okay, so let us focus on this particular part. I know this particular part was written or it is drawn in the very first side, but deliberately I am using this particular sequence of seed development, okay, which is written on this side. Pollen grains. Let there be self-pollination or cross-pollination. Pollen grains are there, and definitely after maturity, when it forms a typical pollen tube contains two male gametes. Okay, they form a pollen tube. I need not to say how pollen tube is formed. Then fertilization in ovule. Fertilization in ovule, when that particular pollen tube reaches up to the micropyle. Try to recall the structure of anatropous ovule. So this particular end through which the pollen tube passes, so it is called as micropyle. So this micropylar end, it always allows that particular pollen tube to enter in. There are several mechanisms, like a typical tube is filled with liquid which is released by the synergid cells, that is sister cells. So they always help that particular pollen tube to burst. When it bursts, the two male gametes which are present, they move inside. The very first male gamete, that very first male gamete, it fuses with the egg cell. When it fuses with the egg cell, it forms a zygote, and zygote further forms an embryo. This is second male gamete. Second male gamete, it fuses with the secondary nucleus. Now it forms primary endosperm nucleus. Primary endosperm nucleus, repeatedly it undergoes mitotic division. It undergoes mitosis. When it undergoes mitosis, it forms actual endosperm, which is nutritive by nature. This nutrition is always required by an embryo, developing embryo. So this is how the overall sequence of seed formation is written here. There are two types of seeds. First one monocot and second one dicot. We are going to learn that particular structure in detail. But before that, I would like to tell you, you have your notes. Yes, I have them. You have your notes, and on sheet number 4 and sheet number 5, the monocot and dicot structure is given. So I want you people to refer those notes and refer them simultaneously. Your NCERT book doesn't give much about the seed, and as I mentioned in the beginning, for this particular chapter we are strictly going to follow the NCERT sequence and NCERT content. But for this particular seed part, we are going to increase our spread by little 10%, say 10%. So this will help you to understand whatever the things are there in standard 12 or in further chapters, that those things you will learn little earlier. Okay, at least you will get some introduction to the next topics. Okay. So this particular session, we are going to start with dicot structure, then monocot structure, and we are going to have germination types. And germination types, yes, there are several types. You are going to learn them. You are going to learn them in plant growth chapter as well. But here we want that you people should understand what is epigel type of germination, what is hypogel type of germination, what is vivipary in the plants. आता vivipary म्हटल्यानंतर आपल्याला animals आठवतात, तसं नाही. We do have certain examples like mangroves. They show vivipary. Okay. I think seed structure, that is monocot, dicot, then germination types and vivipary. That part we are going to learn. Let us begin with the very first part, that is, what is seed? What do you mean by seed? Seed is nothing but the seed coat, outer covering, protective covering it requires, because with the help of seed coat, seed can remain dormant for several years. So it is possible. This is seed coat, this is endosperm and embryo. What do you mean by endosperm? Yes, that endosperm is required by the typical plant. In case of dicots, the endosperm is totally absorbed in the cotyledons. This endosperm is totally absorbed in the cotyledons. In case of monocots, the endosperm is prominently present in the separate chamber, I can say, separate compartment. Try to imagine both the seeds. Try to imagine a typical pea plant, pea seed. सध्या आपल्याकडे मटार भरपूर येतात घरी. सो मटारची शेंग असेल ती उघडा, त्याच्यामध्ये सीड असेल, मार्जिनल प्रेझेंटेशन आहे, आठवते? मार्जिनल प्रेझेंटेशनचं एक सीड काढा. Simply remove the seed coat, gently remove the seed coat and open the cotyledons. You will find some outgrowth over there. So that is nothing but the embryonal axis. What are the things I have drawn here is the ideal diagrams or the schematic diagrams, I can say. In many cases or almost in all cases, you will find embryo is covered in typical coiled manner. If it is present in shrunken manner, doesn't matter. It is an embryo. Okay? आपण मटकीला मोड आणतो. मटकीची उसळ बनवतो ना आपण. मटकीला मोड आणतो. मटकीला मोड आणल्यानंतर ते वरचं जे टर्फल असतं ना, which is greenish brownish in color. So that particular outer covering is nothing but the seed coat. The part which you open, which is greenish yellow in color, that is cotyledon, and whatever the projection like structure you find, so that is radicle, that is a part of root initiating part it is. And simultaneously the plumule, which is leaf like structure, that particular structure is typically what I can say, it is hidden inside the cotyledons. Okay, so this much part you should understand. Similarly, if you take a typical maize grain, maize is a monocot. If you take a jawar grain, so that time you can say I cannot find two cotyledons. Okay, there are no cotyledons. Actually, cotyledon is present, but it's a single cotyledon which is present inside. Then this particular section we are going to learn, and in that section we will come to know how monocot seed is arranged beautifully by the nature. Okay, so let us get back to the dicot seed. Let me tell you the parts of dicot seed first. So these are the two cotyledons. I have opened a seed, and these are the two cotyledons which are present here. Second, this central part, which is axis, it is called as embryonal axis. Okay, so this is embryonal axis, and it has four parts. This embryonal axis is present, and there are certain points at which the cotyledons can attach. The point at which the cotyledons they attach, so it is called as first node. It is called as first node. Then there are two parts, epicotine and hypocotyl. The part which is above the first node, it is called as epicotyle, and the part which is below the first node, it is called as hypo. Epi above, hypo below, cotyle, cotyledon. Now think about plumule and radicle, the part which is responsible for giving rise to a typical plant or shoot structure. So it is called as plumule, and the part which is giving rise to root system, it is radicle. So plumule and radicle. Now I'll modify my statement. This is first node, this is plumule, this is radicle. Now, the part which is present between first node and plumule part, it is called as typical epicotyle. The part which is present between radicle and first node, it is called as hypocotyle. So you need to understand what is epicotyle, what is hypocotyle, because during germination types we need to understand the typical epicotyle and hypocotyle behavior to understand epigl or hypogl type of germination. Okay, so I think this part you have studied or you must have understood. Okay, once again let us revise them. This is embryonal axis, this is cotyledon part. There are two cotyledons, hence it is dicot. So this particular part, dotted part purposely I have shown, because whatever the endosperm is available, free endosperm is not available in case of dicots. So that is all absorbed in this particular cotyledon. त्याला जे आपण द्विदल धान्य म्हणतो, त्यातलं जे एक दल आणि दुसरं दल जे असं असतं. सो याच्यामध्ये तुमचे सगळे टिपिकल कॉटलिडॉनमध्ये एंडोस्पर्म हा सगळा स्टोअर केला आहे. So this is typical seed structure. So let us have a pause and then we'll start with the next part. I'm sorry for the disturbance caused, but once again, this is cotyledon in which the endosperm is absorbed. This particular cotyledon, when it shows its independent presence, that time we should understand it is fully filled with the endosperm. Actually, an endosperm is absorbed. So don't worry about the nutrition which is required by this embryo, whether it is getting or not. Definitely that embryo will get its nutrition, because there are certain mechanisms by which the endosperm which is present, so it will what I can say, it will make itself free, complex structure to simple structure. It will get converted in such a way that it will be easily absorbed, it will be easily usable by the embryo, developing embryo. Now many times you must have seen that seed is growing and seed comes out of the soil, and there are two cotyledons which are present, and between them a typical plant is growing. So this structure you must have seen. Typically in case of tamarind, चिंचेचं झाड असतं, त्याच्या खाली तुम्हाला या सगळ्या गोष्टी दिसतात. या टिपिकल स्ट्रक्चरला हा जो प्रकार आहे कॉटलिडॉन आणि हा जो प्रकार आहे, तर हा न्यूट्रिशन देत असतो. Ultimately, when that particular shoot part it develops into a green part, it means when photosynthesis starts, that time these cotyledons they wither down, they wither off. What do you mean by wither off? So they fall down. When they fall down, means whatever the content is there, whatever the endosperm is there, so it is fully utilized. So this is a typical structure of dicot seed. I think we have covered this much part. Once again, I can take you to the embryonal axis, because you need to understand this very well. So this part, the notch part. So now I'll simply put an arrow, small arrow over there. So this is first node. Why it is called as first node? Now let me tell you this particular structure. When you find like this, if structure is growing above the soil, small green part is above, and these two cotyledons which are now greenish, little pale yellow or greenish in color, through which the entire endosperm is absorbed by the developing embryo. So now they appear like a leaf, hence it is called as, it's a node, very first node. Do you understand what is node and internode? Now you can understand why this particular part is called as first node, because to that point typical dicot cotyledons are attached. Hence it is called as first node. After this, I would like to focus on this much part. Not entire part, but this much part. What do you think? What is that? It is epicotyle. The part similarly which is present between radical and first node. It is called as hypocotyl. This much part I can say radical. The radical always grows first. You remember this sentence. I can use this sentence while framing the MCQs. You should remember this. Radical always grows first. If you put seed on a soil and if you allow that seed to absorb moisture sufficiently, then the very first part which grows down in the soil, that is positively geotropic, is radical. Always remember. So unless and until root establishes itself, you cannot expect that plant is growing. Unless and until roots are fixed, you cannot expect that plant is growing. Okay. Hence this particular part radical. So I am writing here R, radical, and this is plumule. So this plumule and radical, they are growing independently with the help of available endosperm in the seed cotyledon. Once again, you can go through this. If you want, you can pause the video and definitely you can understand. So what I have written on the board, revise it, simply think about it, close your eyes, think about it, and then let us move to the next part. That is all about the actual dicot seed, how it appears externally and how this monocot structure is showing some resemblance, but there are so many differences in the structural arrangement. Now you need to understand what is dicot structure or dicot seed structure and how it appears externally. For example, we are considering the gram seed. तुम्ही घरी आहात म्हटल्यानंतर तुम्हाला एक टिपिकली सीड जो असतो तो सीड तुम्हाला कुठलाही घेता येईल. घरी आपल्या मटार असतात ते तुम्ही बघा, किंवा हरभरे असतात ते बघा, किंवा कुठलाही काबुली चणा असतो छोलेवाला तो तुम्ही बघा. ते स्ट्रक्चर तुम्ही ऑब्झर्व करा. त्यामध्ये काय करता येईल, सिम्पली होल्ड दॅट सीड इन युअर हँड and try to focus on these particular content. So the part which I have shaded, so that is nothing but the outer coat, that is testa plus tegment. Deliberately I am using this word. Seed coat is nothing but the testa and the tegment. Yeah, I have written here. The whatever external part you see, so that particular part which is rough by nature, we show different types of colorations. So that is a typical testa. And when you open it, if you remove the outer seed coat gently, so the part which is present between the cotyledon and the seed coat or the testa is called as tegment. Usually tegment, it appears lustrous by nature. Why it appears lustrous by nature? Because so it is showing its different appearance. So it shows its independent existence. So definitely this testa plus tegment, it forms a seed coat. You cannot easily differentiate between testa and tegment because they are fused. The outer side, that is testa, and the inner side, that is tegment, which jackets the cotyledon. Always remember, consider this part. So this particular part, if it is nothing but the seed coat, the outer part which you can see here is testa. The inner part which is fused with this, is it possible for me to remove this inner skin differently or independently? If I remove it, it is not possible. So this particular thing is fused. So this is fused structure. This fused structure, similarly, it appears in the seed case. So this outer seed coat is testa plus tegment. Now, this particular hilum is the typical attachment point. I am not talking about this particular end notch, a typical pointed structure. I am not talking about that. Before that, somewhere here, the hilum is present. What do you mean by hilum? In case of Anatropous ovule, I had mentioned about hilum and raphe and certain things. So hilum is nothing but the typical seed attachment point. It's a seed attachment point with ovary. So ovary, it always gives a space for seed to show some attachment. So this attachment is, it leaves a scar mark. This scar mark is popularly called as hilum. Just above the hilum, there is presence of micropyle. Now you should try to recall the basic concepts of ovule. Let there be anatropous, amphitropous or orthotropous ovule. There is always a presence of micropyle. What do you mean by micropyle? Micropyle, it's a typical opening in the ovule through which the pollen tube passes. So when this particular micropyle, it shows minute aperture, minute opening. So that minute opening, it remains there throughout the seed. It is not possible for a seed, it is not possible for nature to seal the everything, because there is going to be a necessity of entering of water for germination. So this particular micropyle is present just above the hilum. This is micropyle and this is hilum. Now tell me, what do you think about necessity of this micropyle? Minute aperture. What do you think? What is the necessity of this aperture? First of all, when you soak the seed in water, that time water enters. Water enters into the seed through micropyle. Water never enters into the seed through seed coat. Seed coat madhun जात नाही. Testa, tegment, if they are fused together, they never allow water to enter. But when this particular water enters through the micropyle, slowly the imbibition occurs. Fugna jala म्हणतो आपण. So that imbibition occurs. When the swelling occurs, that time you take out the seeds and you allow it to germinate. So this can be done, this experiment can be done easily in the home. First take some chana seeds, okay, or any other chana, kabuli's chana or chole seeds you can take. You can observe the things carefully. Try to remove the seed coat. It will not be possible for you to remove the seed coat, but definitely after imbibition, after soaking them in the water overnight, you will be able to remove them because it will become soft. So soak them overnight and take out, allow that water to drain. Don't make them very dry. Simply wrap them in a cotton cloth and allow that particular cloth to provide certain moist environment to the developing seeds. There will be certain chemical reactions. There will be, I can say, there will be so many chemical reactions. There will be so hormonal actions through which the germination or the radical formation will initiate. So whenever we talk about a seed, a dry seed, always remember there is a presence of everything. Cotyledons are there, embryonal axis is there, endosperm is stored in the absorbed form, everything is there. But at appropriate time, this particular micropyle will produce a typical radical out. When the radical moves out, it tries to go down in the soil because it is positively geotrophic. Simply put a typical wet seed in a soil and observe. Whatever the white part that comes out, that is radical, it directly goes into the typical soil. Why it goes into the soil? Because it's a nature, it's a tendency of that particular radical. It is positively geotrophic. It moves in the direction of soil. It goes deep down because it needs to establish itself to support the entire plant. So this happens, and then lumen goes out. The food from the food from the cotyledon, it is used, and then the plant grows. So this is the actual behavior of any seed, a typical dicot seed. Now think about the typical endosperm. What do you think about the endosperm, which is present here? This proper endosperm pouch or pocket is present with the monocot seed only. To talk about monocot seed, but I would like to make you aware that read about this endosperm. Why I have written this particular endosperm word and why I have not written the endosperm word over there in the dicot seed? Because this particular structure is a typical monocot seed structure. For example, we have taken an example of maize. This is maize plant. This is maize seed. Let us take a section. This vertical section we have taken. So you may say that that particular maize seed is so small, then how it is possible to take a section? Let me tell you, the technology has advanced in such a way that even you can easily observe the various neurons in the Drosophila brain. The seed is typically visible, but even in case of Drosophila brain, you can observe the neurons under microscope. It is possible. So this scanning electron microscope and other microscopes are easily available, and there are certain agencies which regularly carry out such type of exercises of sectioning and identifying the structure. So that is the different thing, but definitely you can make a typical career out of this. It is possible. Now focus on this particular structure. This particular structure, monopod C structure. Just try to identify the labels. Identify the labels. So this is a typical seed coat. Popularly it is called as hull. Then alluron layer. Don't worry about the various layers and the various terminologies. Then coleoptile. Then plumule, radical, coleorhiza, cotyledon, and embryo. Purposely, I have not put any arrow for the embryo. Okay. Now, consider this as a single cotyledon structure. When we call it as monocot, means monocotyledonous structure. So this monocot structure is just because of this particular name. I will highlight the thing. So this particular part which I am highlighting, it's a cotyledon. Hence it is monocot. Single cotyledon you can find. There are two compartments actually. There is actually a layer of epithelial tissue which is present here, but I am not showing that particular structure. You can refer your notes, page number, sheet number 5. Okay. So you can refer that notes with me. This particular compartment is nothing but the true compartment for endosperm storage. What do you mean by true compartment? This endosperm is separately available for a seed to grow. In case of dicots, free endosperm is not available. Hence, this particular seed is called as albuminous. Albumin. It's a endosperm. Albuminous seed. I'm sorry. Albuminous seed. Dicot seeds are popularly called as non-albuminous or exalbuminous. 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 हे शब्द खूप नवीन आहेत. हे खूप शब्द लक्षात ठेवण्याकरता थोडे कष्ट करावे लागतील. हरकत नाही. थोडं तुम्हाला शाळेमध्ये हे झालेलं असेल. मला तरी वाटतं असं. So this particular albuminous seed, that is endospermic seed, shows prominent presence of endosperm. This particular endosperm is jacketed in the alluron layer. What do you mean by alluron? Alluron, it's a special layer which is protein rich layer, and it jackets the typical endosperm, and there are several chemical reactions they are taking place at the time of germination. So this alluron layer plays an important role in the overall life of a seed. Okay, so this is alluron layer. Now think about this lower part. What do you mean by this lower part? This lower part, actually it contains a cotyledon which is single. Think about this particular depression. Actually the khaddatla bhag. So it appears like this. So whatever the arrow like projections you are seeing here, they are made on this particular structure. So this central part is this, a little depression part. I cannot show it with the 3D markings, but try to understand that particular part is having a light depression. Thoda sa खोल असतो. So this particular plumule, it grows into a typical shoot, and radicle, it grows into a root. Similar to that of dicot, radicle grows first and plumule grows later. But this particular plumule and radicle, they have a typical protective covering. The layer which is present above the plumule, it is called as coleoptile. The layer which is present above the radicle, it is coleorhiza. Rhizoids, root, rhiza, coleorhiza. लक्षात ठेवण्यासाठी सोपं आहे. Radicle, coleorhiza, R. आणि plumule, that is coleoptile, P. Coleoptile, plumule, that is shoot system. This is plumule, this is radicle, this is coleoptile, this is coleorhiza. Now, हे सगळे मेकॅनिझम आहे. हा कोणासाठी आहे? त्या एम्ब्रिओसाठी आहे. आता एम्ब्रिओ कुठे? एम्ब्रिओ दिसतोय का कुठे? This particular part is nothing but the embryo. इथे लक्षात मुद्दा म्हणून मी हे लेबल लिहून ठेवलं होतं, पण त्याला ॲरो दाखवलेला नव्हता. त्याचं कारण काय हे तुम्हाला लक्षात यावं, कारण डायकॉट सारखं हे सिंपल स्ट्रक्चर नाही आहे. एम्ब्रियो ॲक्सिस असेल आणि 2 कॉटलेडॉन्स असतील, हा काही टिपिकल स्ट्रक्चर नाही आहे. दिस पर्टिक्युलर स्ट्रक्चर इज क्वाईट युनिक, and you need to understand uniqueness of this particular seed structure. Okay, once again we'll revise the monocot seed or albuminous seed. Okay, this is a typical maize plant seed. Okay, Zea maize. Zea maize, it's a scientific name of maize. तुम्हाला आता लक्षात ठेवायला हरकत नाही. This is Zea maize and this is a typical seed. We have taken vertical section out of it. ते मधोमध आपण उभंच्या उभं कापलंय. आपण त्याच्यानंतर काय दिसतं हे असं दिसतं. हे लक्षात ठेवा, खूप सोपं आहे. Now This outer layer, it is called as hull, H-U-L-L. It is called as hull. That means seed coat and fruit wall is fused. Caryopsis type of fruit आठवते का? नहीं आठवत? Presentation में. Caryopsis. It's a fruit type in which typical seed coat and fruit wall, it is fused. It's a single seeded fruit, Caryopsis. Grains असतात सगळे, monocots. It's a typical characteristic of monocot, Caryopsis type. So this particular seed coat and fruit wall, together they form hull. And this particular type of arrangement is always seen with the Caryopsis type of fruit. So this is Caryopsis type of fruit. आपण याला seed म्हणलेलं आहे, but it's a fruit. Okay. Now think about this particular major section, that is endosperm content. आपण जो म्हणतो ना स्टार्च प्रकार असतो, that is nothing but the endosperm. आपण कॉर्नफ्लोअर आणतो घरी, कॉर्नफ्लोअर काय असतं? हेच असतं, सगळा स्टार्चच असतो. म्हणून शक्यतो आपण म्हणतो ना की एक मैदा प्रकार खाऊ नये. खूप स्टार्ची प्रकार असेल ना तो चांगला असतो. इट इज नायदर गुड फॉर युअर हेल्थ, नायदर फॉर युअर इंटेस्टाईन, नॉर फॉर युअर थिंकिंग ॲबिलिटी. जेवढा मैदा जास्त तेवढी त्याला डायजेस्ट करण्याकरताची एनर्जी जास्त, आणि जेवढी एनर्जी त्यात जास्त तेवढा इथला जो काही पार्ट यूज करायचा आहे तो तुमचा होणार नाही. सगळं डायजेशनवरती तो एनर्जी खर्च करेल. त्यामुळे अवॉइड बिस्किट्स अँड ब्रेड्स अँड अदर थिंग्स विच आर मेड अप ऑफ मैदा. त्यामुळे हे कुठलेही एंडोस्पर्म प्रकार जो आहे तो फार खाऊ नये. सो दिस इज टिपिकल एंडोस्पर्म and which is present in the aileron layer. It's a jacketed layer. It is not any specialized layer. It's a aileron layer, simple aileron layer, which is it's a specific protein actually. This lower part is showing presence of radicle and plumule along with the protective coverings, coleorhiza for radicle and coleoptile for plumule. प्रोटेक्टिव्ह कव्हरिंग आहे ती नेचरची अरेंजमेंट आहे. तुम्ही असं म्हणाल की यात का आहे आणि यात का नाही आहे, बट आय कॅनॉट इझिली से. पण त्याच्यामध्ये खूप रिझन्स आहेत, खूप व्हेरिएशन्स आहेत. एक प्रश्न होता, एका स्टुडंटने केला होता, चांगला प्रश्न होता की तुमचा हिबिस्कस, जास्वंदीची 2 फुलं त्यांनी ठेवली होती आणि विचारलं होतं की एकाचे पेटल्स लहान आहेत, एकाचे पेटल्स मोठे आहेत, एकाच झाडावरचे, ह्याच्यामध्ये कसं काय? देर आर सो मेनी फॅक्टर्स विच अफेक्ट्स द नॉर्मल ग्रोथ ऑफ फ्लावर. समजा त्या ब्रांचपर्यंत जर का न्यूट्रिशन आलेलंच नसेल पुरेसं, इट विल नॉट शो दॅट मच ग्रोथ. एखाद्या ब्रांचमध्ये कॉम्पिटिशन जास्त असते, नंबर ऑफ बर्ड्स इफ देर आर ऑन द सिंगल ट्विग, then definitely the food will get distributed. So this will affect the overall growth of the flower. It is possible. या नेचरमध्ये कुठली इम्पॉसिबल अशी गोष्ट नाही, सगळ्या गोष्टी पॉसिबल आहेत. देर आर सो मेनी कॉम्बिनेशन्स, देर आर सो मेनी परमिटेशन्स विच आर पॉसिबल. ओके, सो दिस इज प्रोटेक्टिव्ह कवर. दिस सिंगल स्ट्रक्चर इज cotyledon, and this part somewhere here, easily it cannot be seen. Just for your understanding, I have shaded this much part. So somewhere here the embryo is present. So in both cases embryo are present, in both cases plumule and radicle are present. But here in case of monocots, hull is present, outer coat, seed coat, and this particular aileron layer is present just to protect and store the endosperm. So this particular idea about monopod seed you have got. Now I would like to take you to the various germination types, but I think before that, okay, we'll go to the germination types. Certain things I will draw on the board, and then we'll go for the presentation. Okay, till then you should be ready with your extra noting sections. Wherever you are writing them, तुम्ही जिथे कुठे लिहित असाल ते तुमचं सेक्शन रेडी ठेवा अँड हॅव सम कलरफुल पेन्स सो यू कॅन इझिली label the things and you can easily write a comment about it. Okay, get ready. The last part which we need to understand is epigel and hypogel type of germination. Already you have learnt about embryo axis. I'll draw the embryo axis. I'll draw the embryo axis for common for both type of germinations. Now understand this embryo axis. This is first node. Now I'm not going to write entire description of this particular embryo axis. So this is epicotyle, this is hypocotyle. Plumule radical easily you can identify. Polarity you are well aware about. Now think about this particular embryo axis. Now we are going to talk about this epigel type of germination. Epi above, hypo below, gel means ground. It is related to the ground. Epigel germination, that is germination which occurs above the soil, in which the cotyledon moves up in the air from the soil. If you sow it in the soil, at the time of growth, it will move out with the cotyledons and then it will start flourishing. For example, suppose this is soil layer, and if you put a typical a seed over here, what will happen? The radical will grow first. Okay, so this radical, if it is growing, then you can expect that now there will be a development of plumule. Slowly the plumule will grow like this. Now understand the end result of this particular epigel type of germination is somewhat like this. This is soil, this is root system which is well established. So this is a typical plant which is growing, and shoot is also growing. So this cotyledon that moves up. Now here in this particular case, when we expect seed to grow upward direction, that time this particular hypocotyle part, that elongates. So this is epigel germination, that is germination above soil, and in this particular case the hypocotyle part elongates. So this elongates, this widens, this stretches up, and then it moves up, and this particular part which shows attachment of cotyledon, so that grows up. This epicotyle part remains in the same length, it never changes. The length changes for this hypocotyle part. So this hypocotyle elongates and this epicotyle remains same. Now this particular type of germination shows presence of cotyledon above the soil after germination. So this particular germination will show the leaves which are attached to the first node and they are out of the soil, they are present in the air, and somewhat greenish color it appears there in the seed structure, that is Cotyledon structure. So this particular greenish structure, it helps in initial photosynthesis process. It prepares some food for the newly growing leaves. We cannot expect that newly primordial leaves will grow like anything and they will perform photosynthesis and the plant will grow. No, initial support is always required for any plant. So this green colored cotyledons, initially they are creamish in color. Now if they are growing up, so they will change their color and they will become green for a certain period. Then they will perform photosynthesis and this particular growing plant will establish itself. Once the leaves are growing, once they establish it themselves, then these cotyledons, they will wither off. Wither off means they will fall down. There won't be any photosynthetic content or anything. So this part will fall down. Hence, this particular structure is popularly called as the embryonal leaves or the first leaves. So these leaves, they appear green in color initially. When they go up, they appear green in color. Hence they are called as first leaf. First leaf means the Cotyledon structure. Okay. So this is epigel type of germination. Let us think about hypogel type of germination. Hypo means below, gel means soil level. Once again, I draw a typical soil layer, a typical seed. Now the root system will establish itself. Then plumule will start growing. Plumule will start growing, and then there will be not immediately, but somewhere here you can expect there will be a leaf formation, and this is how the plant will grow. The seed, cotyledons, it will remain below the soil, opposite to that of epigel. The seed will remain in the soil, and this shoot part, the plumule, will grow up and it will form entire shoot system. It is possible, yes. This type of variation, this type of magic is possible in the nature. तुम्ही म्हणाले हे तर किती कॉमन आहे, याला मॅजिक म्हणण्यासारखं काय? तुमचा आउटलुक आहे. आम्हाला ते अजूनही मॅजिक वाटतं, कारण इतके त्याच्यात फॅक्टर्स आहेत की ज्याच्यामध्ये काही चेंजेस होऊ शकतात. तरी सुद्धा they never leave their original behavior. They always follow their path. They always follow their orders to grow. ओके, त्यामध्ये कुठेही चूक होत नाही. इतकं परफेक्शन त्या नेचरमध्ये. आपल्याकडे होतं का? आज फिजिक्स करायचं म्हणलं तर मग कंटाळा येतो, केमिस्ट्री करू. केमिस्ट्री करायला घेतल्यानंतर वाटतं की आज नको, जाऊ दे, आज पिक्चरच बघू. बरोबर, पिक्चर बघायचं नसेल तर नको, आज एखादी वेब सिरीज बघू. हे जे सारख्या चेंजेस आणि ह्या गोष्टी होतात त्या आपल्या बाबतीत होतात. तिथे फिक्स पॅटर्न असतो, तिथे कुठे कंटाळा नसतो. केमिकल रिॲक्शन्स असतात, हॉर्मोनल ॲक्शन्स असतात, त्या होतात म्हणजे होतातच, आणि त्याच्यातच खरी मजा आहे. हे नेचरला माहिती आहे. नाउ let us get back to the hypogel type of germination. This seed remains below the soil. When it remains below the soil, this particular part will wither off in the soil itself. असं नाही की ते बाहेर येईल आणि पुन्हा विदरिंग होईल. So it will get degenerated over there in the soil itself. Okay, so this particular behavior where you can expect epicotyle part elongates. Hypocotyle part remains as it is. Here in case of hypocotyle, the epigel, hypocotyle elongates, epicotyle remains as it is. थोडंसं वेगळं. हायपो मध्ये एपी इलॉंगेट होतं आणि एपी मध्ये हायपो इलॉंगेट होतं. सोपं आहे, लक्षात ठेवायला सोपं आहे, पण थोडा वेळ लागेल. कमांड येण्याकरता वेळ लागू शकतो. and that is really normal, really very normal. Hypergl. So this type of germination is hypogl. So we have seen this epigl and hypogl. We are not entering into the details of hormonal action which occurs there while germination occurs. Okay. This concept, vivipary, we are going to learn. Very brief introduction I will give you. What do you mean by vivipary? In case of plants, suppose there is a fruit, consider a mango. Imagine a mango. Now imagine whatever the fleshy part is there, consumable part is there, so somehow it is not affecting the growth of plant or growth of new seed. Okay. Imagine a typical seed grows which is present in the endocarp, stony endocarp, जी कोय असते, त्याच्या आतमध्ये बी असते. So that particular seed is growing and it produces plumule and it produces radicle, and now it is ready for the germination. Ready plant is there, so ultimately it will fall down and it will grow like the original plant. So it is possible. So this particular case occurs in case of mangroves maximally. There are certain families in which this particular vivipary is common. So vivipary means seed germinates in fruit itself. Seed germinates in the fruit itself. What do you mean by germination? Producing radicle out, then producing plumule which grows upwards or which grows in the direction of sun. Okay, so this occurs. So this is plumule and radicle grows fruit at appropriate time. At appropriate time, newly growing plant, that is, which is growing from the fruit, so that detaches from the fruit itself and it will fall down and it will establish itself. So this particular behavior is quite common. This type of behavior is quite interesting to study. So I think we can provide you a couple of links about the same, but you can simply Google the things, go to the YouTube and try to search for vivipary in mangroves. So there are so many beautiful videos available, those you need to watch actually. So go through them and easily try to understand the concept of epigl germination and hypogl germination and vivipary. Now I would like to take you to the screen, and before that there are a couple of announcements. First thing is that I would like to appreciate and congratulate those students, those who have taken efforts to collect the pictures or click the pictures, photographs on a field. I think they must have gone through the nearby areas and they must have clicked the photographs. अतिशय सुंदर पद्धतीने त्यांनी फोटो काढलेले आहेत आणि काही जण बहुतेक तुमच्यातले नर्सरीमध्ये पण गेले होते, हरकत नाही. एकाचं तर सुंदर कॅक्टसचं कलेक्शन आहे. ओके, मी मुद्दामून नावं घेत नाही आहे, आमच्याकडे लिस्टमध्ये नावं आहेत, पण काय होतं, नावं घेतल्यानंतर मग बाकीचे म्हणतील की आम्ही थोडं केलं तरी आमचं नाव घ्या. तसं नाही, आपल्याला काम केलं पाहिजे, कुणाचं नाव इथे सांगितलं जावं म्हणून काम करू. तुमच्याकडनं काम करा आणि इंडिव्हिज्युअली आपण मेसेजने तुम्हाला कम्युनिकेट करतो. ज्यांना मी मेसेज केला आहे त्यांच्या लक्षात येते. आमच्याकडून थोडा त्याला मागे पुढे होऊ शकतं, कारण हे सगळं शूटिंग, रेकॉर्डिंग, तुम्हाला आजूबाजूला आता काही आवाजही येत असतील, हे अनेक फॅक्टर्स असे आहेत की ज्याच्यामध्ये हे शूटिंग चालू आहे. तर हे सगळं मॅनेज केल्यानंतर प्लस ॲडमिशनचे काही आता लोड्स आहेतच, तर हे सगळं केल्यानंतर ज्यावेळेस रात्री आम्हाला वेळ होतो 11-11:30 नंतर, त्यावेळेस आम्ही तुम्हाला रिप्लाय करत असतो. अतिशय चांगल्या पद्धतीने काम झालेले. याच पद्धतीने तुम्ही तुमचा पेस ठेवा. जर्मिनेशनचे एक्सपेरिमेंट्स तुम्ही घरी करू शकता. घरात आपण जे जे खातो ना, मूग, मसूर, चवळी, शेंगदाणे, छोले, राजमा, हे सगळं आणा. सगळं वेगवेगळ्या वाट्यांमध्ये भिजत ठेवा. भिजवून झालं ते एक रात्रीकरता भिजवायचं. एक जनरली 8 तास लागतात भिजायला. एक रफली सांगतो आहे तुम्हाला, 8 तास लागतात. आणि मग ते काढून घ्यायचं आणि ते छोटे रुमाल घ्या, कापड असतं ना त्याच्यामध्ये ना ते रॅप करून ठेवा. एकदम गच्च पॅक नाही करायचं. अलाव दॅट पर्टिक्युलर सीड टू ब्रीथ. त्याचं ब्रिथिंग झाल्यानंतर त्याच्यामधून तुम्हाला ते मोड येताना दिसतं. मग त्याला एकदा मोड आले ना की पुट दॅट पर्टिक्युलर सीड विच इज जर्मिनेटेड इन टू द सॉईल. आणि सॉईलमध्ये टाकल्यानंतर बघा काय होतं, कसं ते ग्रो होतं. दुसरा एक्सपेरिमेंट असा करा की एक मोठा ट्रे घ्या, त्याच्यामध्ये प्रॉपर एवढी सॉईल घ्या. इट शुड बी ऑफ 3 इंचेस, डेप्थ शुड बी ऑफ 3 इंचेस अँड सो सम सीड्स. एक रांग तुम्ही शेंगदाण्याची करा, एक रांग तुम्ही मटारची करा, एक राजमा, एक चवळी, असे वेगवेगळे करा. आणि त्यातले बघा कुठले एपीजीएल आहेत आणि कुठले हायपोजीएल आहेत. सोपं आहे. ज्याचं कॉटलिडॉन वरती येईल दॅट बिकम्स एपीजीएल. ज्याचं कॉटलिडॉन खालीच राहील आणि फक्त प्ल्युम्युल ग्रो होईल, शूट तयार होईल, दॅट इज हायपोजीएल. आणि आता मॅन्ग्रुव्हचं तर तुम्हाला लगेच शक्य नाही. एक तुम्ही करू शकता की हा एपीजीएल हायपोजीएलच्या एक्सपेरिमेंटबरोबर एका वेगळ्या बाउलमध्ये किंवा ट्रेमध्ये दॅट ग्लास शुड बी ॲक्च्युली ट्रान्सपरंट. तर जर का ग्लास असेल, प्रॉपर ग्लास असेल, ट्रान्सपरंट ग्लास, तर त्याच्यामध्ये तुम्ही हे घ्या, सॉईल घ्या आणि त्याच्यामध्ये गहू टाका. त्या गव्हावरती परत थोडीशी सॉईल टाका आणि अलाव दॅट पर्टिक्युलर प्लांट टू ग्रो. सो इट विल गिव्ह यू अ क्लिअर impression about how roots are growing. So you can observe them from outer side. ते सीड्स टाकताना मध्यभागी टाकू नका, काही दिसणार नाही. यू स्प्रेड दॅम अलॉंग द रिम. ॲक्च्युली त्याचे पेरिफेरी जे असेल ते रिमवरती तुम्ही ते टाका आणि त्याच्यावरती सॉईल टाका. वॉटर इट प्रॉपरली. खूप त्याच्यामध्ये खूप सॉगी करू नका, थोडं पाणी टाका आणि इझिली यू कॅन ऑब्झर्व द ब्युटिफुल रिझल्ट्स आउट ऑफ इट. पुन्हा एकदा तुमच्या फोटोची आम्ही वाट बघतोय आणि माझं म्हणणं आहे हे सगळ्यांनीच करावं. सगळ्यांनी थोडं करून बघितलं पाहिजे. ज्यांचं सायन्समध्ये इंटरेस्ट या पद्धतीने वाढेल त्यांचं सायन्समध्ये करिअर होईल. फक्त मला KVPY करायचं आहे, मला ऑलिम्पियाड करायचं आहे आणि फक्त हे असे फोटो काढून आणि एक्सपेरिमेंट्स करून KVPY ऑलिम्पियाड होतं का, डॉक्टर होईल का, ह्याला काही अर्थ नाही आहे. सायन्स ही एक टेंडन्सी असते. सायंटिस्ट हा एक ती कम्युनिटी आहे की ज्याच्यामध्ये त्यांचा स्वभाव वेगळा असतो. मी MSc झालो म्हणून मी सायंटिस्ट आहे असं नाही. मी PhD केली म्हणून मी सायंटिस्ट आहे असं तर मुळीच नाही आहे. PhD तर काही गव्हर्नमेंटच्या रूल्स करता आजकाल केली जाते. आवड म्हणून कोणीच PhD करत नाही. स्केल, सॅलरीज, परमनंट जॉब्स, बरोबर ना. तुमच्या आजूबाजूला असे एक्झाम्पल्स असतीलच. त्याच्याकरता ह्या PhDs होतात. खूप थोडे असतात, खूप थोडे. आणि त्याच्याबद्दल आम्हाला खरंच रिस्पेक्ट आहे की जे आवड म्हणून फक्त या PhD किंवा या रिसर्च सेक्टरमध्ये आलेले असतात आणि त्यांचाच खरा रिसर्च असतो, त्यांचंच खरं सायन्सला कॉन्ट्रीब्युशन असतं. यातलं आम्ही काय केलं आहे सांगा. आम्ही कधीही सेक्शनिंग केलं नाही, कधी एंडोस्पर्म वेगळा तो हायलाईट करायचा प्रयत्न केला नाही, त्याचं स्टेनिंग आम्ही केलेलं नाही. आम्ही फक्त शिकवण्याचं काम करू शकतो, अभ्यास करतो आणि चांगलं शिकवण्याचं काम करू शकतो. पण सायंटिस्ट जे आहेत त्यांनी हे सगळं शोधलेलं आहे. एपिकोटाईल, हायपोकोटाईल तुम्हाला आम्हाला कुठे शब्द येत होते, ते त्यांनी शोधलेले शब्द आहेत. आणि सायन्स तेव्हाच वाढतं ज्यावेळेस तुम्ही थोडंफार एक डेडिकेटेडली काम करा. आणि जसं आता तुमच्यातले काही जण करतात, फक्त ती जी टेंडन्सी आहे ना, थोडं करा पण छान करा. ती टेंडन्सी थ्रूआउट लाईफ तुम्ही ठेवा. तुम्ही बायोटेक्नॉलॉजीला जा, मायक्रोबायोलॉजीला जा, प्लेन सायन्सेसला जा, झुलॉजी करा, बॉटनी करा, स्टॅटिस्टिक्स करा, काही करा पुढे जाऊ. पण तुमची जी ही इच्छा आहे चांगलं काही करण्याची ती नक्की ठेवा. मेडिकलला गेल्यानंतर मी काही बोलत नाही, मग ते सगळं चॅनलाइज होऊन जातं. पण तरीसुद्धा अनेक लोक असे आहेत की जे मेडिकलला गेल्यानंतर रिसर्च ऑप्शनचा विचार करतात आणि तसं झालं पाहिजे. एम्स आणि अनेक हॉस्पिटल्समध्ये खूप चांगले रिसर्च स्टेशन्स, रिसर्च लॅब्स आहेत, तर तिथे काम केलं गेलं पाहिजे. ऑलरेडी एवढे ह्युमन बॉडीज आहेत तुमच्याकडे, तुम्ही इझिली त्याच्यावरती काम करू शकता. हा सगळा प्रकार सांगण्यामागचा उद्देश एकच आहे, तुम्ही सायन्स चांगलं करावं, ऑनेस्टली करावं, सिन्सिअरली करावं. जसं तुमच्यातले आत्ता बरेच जण करतायत, जे करत नाही आहेत त्यांचे काही इंटरेस्ट वेगळे असतील असं समजूया, पण त्यांनासुद्धा जर का वाटत असेल आपण काही करून बघावं तर नक्की केलं पाहिजे. तुम्ही थोडं बाहेर जा, थोडं बघा, पेरेंट्सची परमिशन घ्या, जे काय असतील ते रूल्स रिस्ट्रिक्शन्स फॉलो करा, मास्क वेअर करा आणि प्रॉपर तुम्ही बाहेर जाऊन काम करू शकता. एवढी सिच्युएशन आता नक्की आहे. पण कुठे तुमची रिस्क कुठली घेऊ नका आणि कुठे धोका होईल अशा ठिकाणी जाऊन काम करू नका. ठीक आहे, आता मला वाटतंय आपण प्रेझेंटेशनकडे जाऊ शकतो आणि प्रेझेंटेशन झाल्यानंतर हा टॉपिक संपतो. प्रेझेंटेशनला जास्त नाही, हे एवढा जो पार्ट आहे त्याच्याच थोड्या स्लाइड्स आहेत. ओके, now we are going to start with the actual presentation. So this is all about seed, and here you can easily identify what type of germination it is. Yes, it is Epigl. Here you can find the epicotyle is not getting elongated. Whatever the part is getting elongated is hypocotyle, which is below the first node, and it is between first node and the radicle. So hypocotyle is getting elongated, not the epicotyle. This is seed. Actually we are studying about seed, and these are the newly formed leaves. And so there are 4 GIF images. You can find this particular seed is growing and the radicle grows first, and it always moves in the direction of soil. So it is positively geotropic and negatively phototropic. Very beautiful GIF. Look at the germination. It's a time-lapse photography. See how the things are getting formed. As I mentioned, so this particular Epigl type of germination, see the cotyledons are moving up. The elongating part is this hypocotyle. The epicotyle elongates, but after the proper development, then it elongates slowly, not very fast. So repeatedly you can see this particular GIF. Think about this. Purposely I am taking a pause. Observe the things carefully. See the shrinking behavior of cotyledon, how it shrinks. Initially it is quite healthy, but eventually as this particular part grows, it shrinks. It is shrunken. Let us go back first. So this is radicle moving. This is plumule growing. This particular this was Epigl. Think about this. What type of germination it is? These are the wheat grains. These are the wheat grains. So you can perform this particular experiment in your regular transparent glass. Okay, so don't worry much about the aids which you are using. Simply think about the result which you want. Okay. Yeah, another monocot, that is maize. Earlier was a typical wheat grains. and this is maize grain. Radicle grows first, plumule grows later, it goes above the soil, and whatever the endosperm this particular structure has, so it is utilized. Understood? So let us revise certain things. So look at this particular structure. It's a dicot, paisa misatibon. वाटाण्याचं शेंग असते ना त्यातलं. So if you take a section, so definitely these are the cotyledons. आपल्याकडे ते आपण बटरफ्लाय शेप मध्ये बघितलं होतं, central axis and the two cotyledons. So this is the vertical section and this is storage for endosperm. Entire endosperm it gets absorbed, so no free endosperm is available. Hence such type of seeds are called as exalbuminous seeds, that is dicot seeds. Testa, that is seed coat, the inside of the testa is fused with the tegment. This is plumule or plumula and radicula or radicle. This is hypocotyl, somewhere here epicotyl, and collectively this particular part is called as embryonal axis. Parts of a seed with functions. This particular section is of maize plant, maize seed. I think in your NCERT page number 77 you can find this diagram. So this is small embryo, this is seed coat or hull you can say. Here in case of dicot, testa and the inner tegment, but both are fused, you cannot separate them easily. For your understanding, the schematic drawing has been chosen. So this is plumule, radicle, hypocotyl, epicotyl. These are the cotyledons. Once again, Monocot seed structure. Just observe that I did not to say much about this. Same one. तुमच्या NCERT गंमतशीर डायग्राम्स दिलेले आहेत, तर ठीक आहे त्या बघून घ्या तुम्ही. पेज नंबर 77. Monocot, earlier was dicot, this is monocot. It's a corn seed or fruit you can say. A section has been taken. This is endosperm, this is seed coat, the allurum there is present, the sheath, the embryonic shoot, and the plumule radicle you cannot easily find out. Coleoptile, coleorhiza you cannot see. Here you can observe the things. So this is true endosperm which is present. Then, as I mentioned, there is some presence of epithelial layer. It is present. Then plumule, radicle, coleoptile and coleorhiza, root cap. So everything is present. So we need not to enter into details of this particular structure, but try to understand. Endosperm is freely available. There are certain exceptions in dicots also. Earlier I made a statement that dicot seeds are without any endosperm. Whatever the endosperm is available, it is all absorbed in the cotyledons. So here in case of monocots, endosperm is available, but there are certain exceptions like castor plant which are dicot, but still they have endosperm. So this exception you need to remember. Once again, I can ask this particular question for your next combined chapter test. Which of the following plant will show the endospermic seed, though that particular plant is belonging to the dicot structure? Then you should surely say this particular plant is nothing but the castor plant. So popular example is castor. So we are not going to give you any other example. So castor is quite popular example. Yeah, this is the actual section for monopod seed. It was sectioned like this. This is the endospermic part. Somewhere here embryo, coleoptile, coleorhiza. So this is the typical magic of this particular stain. There are so many stains. A stain which can, the chemical which can actually stain or give color to the protein, which can actually give color to the starch or to the growing tip. Whatever the things are there, there are so many chemical issues pertaining to this. Germination type. This is Epigel, this is Hypogel. So I think you are well aware about the things. In Epigel type of thing, the Hypocotyl elongates. In Hypogel type of thing, the Epicotyl elongates. In Epigel, seed grows above the soil level. In Hypogel type, the seed remains inside the soil. So this is Cotyledon, this is Cotyledon. So it is easy. This is vivipary. See, initially this was a flower. You can find these are the calyx. Now this ripened ovary you can find. So this ovary, that is fruit, will contain some seeds. These seeds will germinate inside the fruit itself, because this particular mangrove plant, it grows in a soggy condition. So it is not possible for that particular plant to waste energy in developing seeds and throwing them away in the mud or in the water to get rotten. So just to avoid that, this particular fruit, it has developed a mechanism that the seeds will germinate inside the fruit itself. This is single seeded fruit, and the part which is growing is nothing but the radical. So this radical part, it grows out from the fruit and it acts like a dart. Suppose this is dart, and suppose this is a typical radical of a mangrove fruit. Okay, so from height, it directly drops down into the soil, muddy soil, in the water. So it acts like a dart. So it grows there, and radical establishes itself, and the plumule grows. So this is how this particular mangrove part grows in the nature. I think we have done with the details and all the things I have conveyed to you about seed. I think you should write the things in the practice book. तुम्ही लिहिण्याची खूप प्रॅक्टिस करा. Unless and until you write, you cannot remember the things. Okay? So I think we'll finish with this, and after this particular session, we are going to start with the actual family studies. Family studies we are going to do in a little different manner. Initially, by practical way, we are going to learn how to study the family. For that, we are going to consider the Malvaceae. Malvaceae examples, Sandra? Malvaceae examples काय आहे? Yes, hibiscus आणि भेंडी, ओकरा, दोन्ही एक्झाम्पल्स. तर हे एक्झाम्पल्स आपण घेणार आहोत आणि त्याने तुम्हाला सांगू की हाऊ दिस पर्टिक्युलर फॅमिली स्टडीज इज टू बी कॅरीड आउट. ओके, या सेशन नंतर तुम्हाला याचे प्रेझेंटेशन्स आणि बोर्ड रायटिंग आलेलंच आहे, आणि त्यानंतर तुमच्याकडे टेस्ट पण आहे. आता टेस्ट जी असेल ती फ्रूट आणि सीड अशी एकत्र असेल, तर ती आय थिंक दॅट टेस्ट नंबर विल बी 6+7. ओके, सो राईट दॅट टेस्ट अँड सबमिट इट इन टाइम. ओके, all the best, study well, stay healthy, stay fit. Okay, bye bye.